Hello, Chem 151 students and other chemistry aficionados everywhere. Welcome to the wonderful world of green fluorescent protein, better known as GFP. While the protein has a name that speaks for itself, its properties and applications are truly something special and something that should be appreciated everywhere. You may be asking yourself, what exactly is GFP? Well, the answer is that it's a naturally occurring protein that was originally found in the jellyfish Aquaria victoria. A Nobel Prize was awarded to Osamu Shimamura in 2008 for the discovery and the development of the GFP protein, although he originally isolated it in the year 1968. Green fluorescent protein has the ability to emit a highly visible light when exposed to certain radiation on the ultraviolet spectrum. GFP is particularly useful for molecular biologists and researchers that are in related biological fields. The reason for this is because it's possible to insert the GFP gene into an organism's genome. The result is a physical trait that appears to glow under UV light. The unique properties of green fluorescent protein are due to GFP's unique chemical structure. GFP is a very large protein made up of about 238 amino acids. The structure is made of 11 beta strands, which form a large beta barrel, which takes a cylindrical shape. At the center of the beta barrel lies an alpha helix. Also in the center of the beta barrel exists a chromophore. This is where the magic happens. The unique chemical structure of the chromophore is what allows for the entire protein to fluoresce under certain conditions. We should all take a moment to thank the chromophore for giving us the GFP we know and love today.
In 1995, UCSD's own Roger Chen published a paper that described an S65T point mutation that increased the fluorescence intensity and photostability of GFP. While GFP had been researched and used prior to 1995, it was arguably Chen's paper that facilitated the widespread use of GFP and its mutants in research. Chen's study allowed for further research to produce GFP mutations which respond to wider ranges of wavelengths and emit wider ranges of colors, allowing researchers to fuse various colored fluorescent proteins to proteins of interest to study them. Since then, researchers have introduced various other mutations to GFP, leading to new iterations of fluorophores of different colors. This image right here showcases a couple different variants of GFP in different colors. These tables briefly summarize a couple different point mutations of GFP. Table 1 shows point mutations that produces different colors of fluorescent proteins, and Table 2 shows point mutations that affect the functional role of GFP, such as increasing the fluorescence or folding efficiency of GFP. Due to GFP's structure, it is extremely stable, allowing it to be resistant against heat, alkaline pH, organic salt, and various other denaturants. In addition, GFP is non-toxic and does not affect the function, biological processes, or mobility of the protein of interest when fused. These characteristics of GFP has enabled its widespread use in cell development studies. Next, we will be discussing a specific application of GFP and its mutants, known as fluorescence resonance energy transfer, which is often referred to using the acronym FRET. FRET is a laboratory technique that employs the use of GFP and its mutants to help identify the proximity of two biological molecules, allowing for the study of protein-protein interactions. First, two proteins or molecules of interest are each tagged with a fluorescent protein. Often, the cyan fluorescent protein and yellow fluorescent protein pair is used as the emission and excitation spectra of the fluorescent proteins overlap. Here you can see a graph that shows the emission and excitation spectra of both the cyan and yellow fluorescent proteins. The excitation spectra for both proteins have been graphed with a dotted line, and the emission spectrum of CFP has been graphed with a solid line. The green area that you can see is the overlap of the emission and excitation spectra of CFP and YFP respectively. After the proteins of interest have been tagged with the fluorescent proteins, Light is shown on the sample, which is absorbed by the protein with the shorter excitation wavelength, which is CFP in this case. The light absorbed by CFP allows CFP to enter an excited state, causing it to fluoresce, which in turn is then absorbed by the fluorescent protein with the excitation spectra with the longer wavelength, which in this case is YFP. This then causes YFP to enter an excited state and emit light as well. This technique allows researchers to identify if the two molecules of interest are within a close proximity of one another. If the molecules are too far apart or blocked by some other molecule or protein, the light emitted by CFP would not be able to reach and excite YFP, causing only one of the fluorescent proteins to fluoresce. This would signal to researchers that the molecules are too far apart and likely not interacting. The interaction can then be quantitatively described by the ratio of donor and acceptor emission intensities. That concludes our video on GFP, or green fluorescent protein. Thank you so much for watching, and we wish everyone good luck on finals and have a great spring break.